Good morning, everyone. My name is Blair Fauché. My wife and I, Elise, have a 13-year-old son, Hunter, with type Usher 1C. I remember my first conference in St. Louis, I believe it was 2013. It was literally in a room maybe a third the size of this here. I remember the heat. It was 110 degrees, forcing us inside for the Saturday afternoon social. I remember walking in as a new parent, just simply being overwhelmed. But I remember leaving on Sunday with a sense of comfort about the team here and what all these people do for this syndrome and the passion they have for this syndrome. With that being said, my South Louisiana connection has given me the opportunity to introduce your next speaker, Dr. Jennifer Lentz. Dr. Jennifer Lentz is an ENT from New Orleans. She is a professor of biocommunications, ophthalmology, and genetics, and is a member of the Neuroscience Center of Excellence at the LSU Health in New Orleans. The goals of Dr. Jennifer Lentz's research are to develop therapeutic approaches to prevent or cure the deafness, imbalance, and blindness associated with Usher syndrome, and to develop novel tests in the clinic that can be used to determine whether a new therapy is working. If you guys please help me welcome Dr. Jennifer Lentz.
Good morning. It's such a pleasure to be here. As a research scientist, this really is one of my favorite meetings that I attend. And the first meeting I attended, I think, was 2014 in Portland, Oregon. So I want to thank very briefly Krista and Julia and the whole Usher Coalition team for the opportunity to be here today to present to you. And I'm going to get myself oriented here for just a minute. Do I control changing the slides with this? Yeah? Okay, thank you for the thumbs up in the back. So today, this screen here is what I'm reading from, and it's very small. So for those that are going to use it next, you might want to bring your own computers, because I can't see it from here. So this morning, I'm going to talk to you a little bit about Usher. If you're new to Usher, what we know about it. And then I'm going to give you just a few brief updates on some Usher research that's ongoing with some examples from research in my own laboratory. Okay, so Usher syndrome is defined as an autosomal recessive genetic disease that causes the loss of hearing, balance, and vision. That's a long definition with some complicated terms. So autosomal means that it affects both males and females equally. And recessive means that you need two mutations in order to have Usher syndrome. One mutation that you get from your mom and one that you get from your dad. The prevalence or the frequency of Usher in the community of all types is about 1 in 20,000 individuals, which means there's approximately 400,000 individuals living with Usher in the world, about 40,000 people in the US, and about 37,000 in Europe. So this is a rare disease. Diagnosis today is still generally made based on clinical symptoms. And this is based on the severity of your symptoms and the age in which they begin or onset. Today, we use genetic testing to confirm that diagnosis of the type and subtype of Usher syndrome that you have. And Celeste Wyman is going to talk more about the process of genetic testing today, later this afternoon. So we typically talk about Usher having 3 main clinical types and about 10 subtypes. We use the number or numbers 1, 2, or 3 to describe the symptoms that you have. And then we also use the letter to note which gene you have a mutation in. So for example, type 1 Usher, there are 5 known genes that cause Usher. So you can have USH1B with mutations in the gene myosin 7A, or USH1C with mutations in the USH1C gene, and so on and so forth. USH2A with mutations in the USH2A gene, and USH3 and 3A and 3B. New to Usher syndrome is a type 4. It's very rare. And also there are atypical forms of Usher. And traditionally, this meant that you have a mutation in a gene that's known to cause Usher, but your symptoms are different and don't fit that group. And Dr. Kenna next is going to tell us more about these rarer types and the atypical types of Usher syndrome. Okay, so what we know about Usher syndrome, the types and the subtypes, can help you better understand the symptoms that you have and what you might be able to expect in the future. It can also help you to talk to your doctor, to your loved ones, to your friends, school, workplace, about what you need now and what you might need in the future. It can also help you to know and seek out or stay informed about research, studies, trials, and new therapies that might benefit you in the future. In the next few slides, I'm going to talk a little bit about the natural history of the different types of Usher syndrome as a whole. So, as a whole group of Usher 1, or a whole group of Usher 2, or a whole group of Usher 3. And this means, for example, if we were to survey all of you here today living with Usher about your symptoms, and then average those results, this is what we would find, which also means they might not represent or explain any one of you individuals. So typically, the sensory neural hearing loss for individuals with type 1 Usher is present at birth, and it's stable. It doesn't typically change over time. It's usually severe to profound, and individuals today receive cochlear implants for their treatment. Individuals with type 2 also are born with hearing loss. It's typically mild to severe, which means that it's a mild loss in the low frequencies and a higher loss in the high frequencies, or more severe loss in the high frequencies. Most of these individuals also benefit from hearing aids. And individuals with type 3 usually have a progressive, mild to profound hearing loss that begins later in life. The treatment for individuals living with type 3 usually begins with hearing aids, but can progress to cochlear implants. The vestibular areflexia, or the balance loss associated with Usher syndrome, is typically present at birth for individuals with USH1. It's mild to severe, And these individuals often benefit from physical therapy to strengthen the muscles that we use for balance. Individuals with type 2 are often asymptomatic, even though they may have some changes to their clinical testing of balance when they go to the doctor's office. Some individuals with USH2 also have episodes feeling as though they're imbalanced. And individuals with type 3, some have a feeling of a loss of balance and some do not. It can also be progressive, from mild to profound. And again, the treatment is physical therapy. The retinitis pigmentosa, or the vision loss associated with Usher syndrome, for individuals with type 1, the onset is typically early adolescence. We usually say around the age of 10, but it can begin as early as 5 or as late as 15. It begins usually with night blindness, sometimes associated with photophobia or sensitivity to bright light, and then progresses to a peripheral vision loss and eventually a central vision loss. Individuals with type 2 experience the same symptoms, but it begins later in late adolescence, usually around the age of 18 to 20. And individuals with type 3, also the same symptoms in course, but beginning even later in adulthood. Okay, so I just wanted to spend a minute to talk about the research path to medical progress. This often begins with observing the symptoms in patients with Usher syndrome in the clinic. We answer questions like, what symptoms do you have? When do they begin? And how quickly do they progress? And this information informs research laboratories to develop a model of Usher syndrome, a model that we can use to study hearing, balance, and vision. So we use cells in a dish or research animals. This allows us to conduct experiments on their eyes and their ears. And we use a whole range of animals in order to conduct these tests. And that's because any one animal is usually pretty good at answering some questions with respect to hearing, balance, or vision, but sometimes not all the questions that we have. So we specialize our questions and make special models for each question. We use this information then and these models to develop new therapies in the laboratory. We validate the drugs in the animals. We test to see if the drugs are working and whether they're safe. We optimize the drugs in the animals. And then, if we're lucky, we get to test them in patients. And sometimes the drugs work as we predicted in patients, and sometimes they don't. Sometimes they work for some patients, but not others. And so the process continues. And we observe again in the patients that these drugs may not have worked in. Maybe we make a new model or design a new drug, and the process continues. And the goal for us in research and medicine is to be able to treat and develop a drug for every individual with Usher syndrome. So this is a typical drug development pipeline highlighting 3 main activities just to get to a clinical trial that allows us to test a new therapy in patients. The beginning phases are research discovery and the development of that drug that we want to test in the clinic, followed by a meeting with the FDA to ask for an investigational new drug clinical trial. and lastly, getting to the clinical trial. So that research, discovery, and clinical drug development phase can be further broken down and can take a very long time. But in general, we're asking, how does the drug work in a model of the disease? What doses does it work at? What cells or proteins or other drugs is it interacting with? Also, we're asking, is it safe? Can we deliver the drug to several animal species and it not harm them? And lastly, who are our patients that would benefit from this drug? And what is the measure in the doctor's office that's going to tell us whether this drug is working? We use all of this information and gather other information about how to manufacture the drug, how we would use the drug with the patients or the clinical protocol, what dose, how many times do we give it, what age do we give it at. We gather all of this information and apply with an application to the FDA to give it a try in patients. And lastly, we get to our clinical trial. And Dr. Levin is going to tell us a little bit more about participating in clinical trials. And so I'm going to switch a little bit and talk about this first phase with some examples from research in my own laboratory. Before that, though, I'm going to talk a little bit about the different strategies that we use to develop drugs for Usher syndrome. I'm just going to pause for a minute. I see some switching of interpreters. All ready? All set? Great. Okay, so broadly, one more minute. In the research field, we can categorize drugs using genetic strategies or non-genetic strategies. For the genetic strategies, we're typically targeting the DNA or RNA associated with the disease. And in non-genetic strategies, we're typically targeting a pathway using small molecules or cells that are associated with the disease. So later this morning, Dr. Jeff Holt's going to tell us more about some genetic strategies that are being used. Susie Trodejo is going to connect with us, we hope, online to tell us about participation in an antioxidant drug trial. And John Scholler and David Gamm are going to tell us about small molecules or cellular models for drug development. So I'm going to focus a little bit on the genetic strategies, which are strategies that we use in my own laboratory. The goal of these strategies ultimately is to increase or decrease protein levels to either slow or halt the disease process. Antisense therapy is a method to modify RNA to either slow or halt the disease progression. It's also known as RNA therapy or ASO or AON therapy. Gene therapy is a method that introduces new genetic material, either DNA or RNA, using viruses that express beneficial proteins in cells. And gene or RNA editing is a method to change the DNA or RNA by inserting, deleting, modifying, or replacing nucleotides. Okay, so you might recognize this drug development pipeline that I presented earlier This is a pipeline that we're using in my laboratory to develop drugs to treat the vision loss or the retinitis pigmentosa or imbalance in Usher syndrome type 1C. The beginning phase, the research discovery, target validation, is complete. We've identified a target, tested it in animals, and right now we are optimizing. We want to know if we can increase the drug activity or increase the number of patients that would benefit from this therapy. We're testing both and comparing antisense targeting and gene replacement. We are also conducting a number of natural history studies with patients that have USH1C to better understand their natural course. So the next two slides are just a little bit of data from our lab demonstrating that we can deliver these drugs to the ears or the eyes of our research animals to restore hearing, balance, and vision. On my left-hand side, your right-hand side, where it says hearing thresholds, you can see in mice that we have delivered the drug. We have lowered their hearing thresholds. That's the blue line in the middle. Lowered from the red line, which are Usher mice that do not have treatment. The middle graph, the open field analysis, is how we observe their balance. The Usher mice run in circles because they have a difficult time knowing where they are in the world. And when we deliver antisense drugs to their ears, they stop circling. Those are the blue bar graphs compared to the Usher mice with the red bar graphs that have not received the drug. And the last graph on my right side, on your left side, is a graph of their vision. And it's the opposite way as their hearing. So for the green line, they have more vision, and we have increased their vision compared to the red line, which is below it. The green line represents Usher mice that have been given ASOs to their eyes, compared to the Usher mice in the red line that have not received treatment. And similarly, we have delivered a gene replacement therapy either to the ears or the eyes and have improved their hearing, improved their circling, and improved their vision. Although you'll notice the vision graph is slightly different with the AAV, and that's because we've only tested it in one mouse. So we're still waiting to see, on the whole, if we deliver it to many mice at different doses, if it will improve vision for all of them. So we're continuing to optimize and compare these two therapies to determine which one we'd like to talk to the FDA about. Our challenges with the antisense therapy is that it requires multiple treatments that continue throughout life. Also, it would only benefit those individuals that have one specific mutation in the USH1C gene. As opposed to the gene replacement therapy that we would hope would only have one treatment for the patient to last their lifetime and be able to treat other individuals with USH1C that have different mutations within their gene. Okay, so there are similar strategies for almost all of the types of Usher syndrome. So I'm just briefly going to end and talk about additional strategies and additional research that's going on in the world today. There's one antisense therapy that is currently being tested in the clinic for some individuals with USH2A. This is the QR421A or ultevursen drug, which is an ASO drug that's designed to target mutations in the exon 13 of the USH2A gene. The treatment goal is to stabilize vision loss. So from the moment you receive the drug, the goal is to stop the vision loss from progressing. There have been 3 clinical trials in the United States and Europe, and right now, currently, the trial has been suspended or terminated because ProQR is looking for a partner to conduct the last clinical trial, which they have now found in Thea, and Andy is here to talk about the next steps to continue this clinical trial. Additionally, today, there are preclinical studies or studies in animals using antisense and gene therapy for USH1B, USH1C, as I mentioned, USH1D, USH1F, USH2A, and USH3A. And lastly, there are a number of natural history studies that are being conducted throughout the United States and in Europe. The purpose of these studies is to collect information about the natural history or the course of a disease in the absence of an intervention. And we want to collect information from the onset of the symptoms, maybe even before they begin, all the way through later stages of disease. The goals are to improve clinical care and to benefit drug development. There are ongoing studies for USH1B, the vision testing. USH1C, my laboratory is conducting for both vision and balance. USH1D, USH1F, USH2A. And all of these studies are in progress and enrolling participants. So in summary, Usher is an autosomal recessive genetic disease that causes the loss of hearing, balance, and vision. It's very much a rare disease. There are 3 main types and 10 main subtypes of Usher. Currently, an ASO drug strategy is being tested in some USH2A patients, and there are natural history studies with USH1B, C, D, F, and USH2A patients. And also there are various genetic drug strategies being tested in animal models of almost all the Usher types. With that, I'll just end with a large number of individuals that helped me conduct these studies, both in my own laboratory and throughout the world. Thank you.